Okay, so today we're moving away from the molecular side of biology, away from the cell level and on to the organ system level. So where we're going to start is with the digestive system. And just to cover some bases, we're going to go over the different parts. So some of this is stuff that you've known since the third grade, and some of the terminology is going to be new to you. So there are familiar parts of the digestive system that you guys already know, like the mouth and the esophagus and the stomach. A new word for you might be something called the alimentary canal. And so what the alimentary canal is, it's really shorthand for the entire path. You know, that one long tube that where food enters and is digested, and then that waste product enters. So the whole thing together, mouth, esophagus, intestines, all that good stuff, together is called the alimentary canal. And so if we look at each individual part, again, some of this you will know and some of this is new to you. We know that the mouth is where we chew the food, and so the purpose of that is to increase surface area on the food to allow enzymes that are going to be acting later to have access to that food to increase surface area. So from there, it has to get from the mouth down to the other, the other parts of the alimentary canal. And so it enters the esophagus, and what the esophagus is doing is actually moving that food to the stomach via a really special movement called peristalsis, and we'll talk more about that in a second. So the next one in line is the stomach, and so this one you're probably very familiar with. The stomach has a really acidic environment. Its main job is to kill pathogens, and this is where many of the first digestive enzymes become very active, and it's also turning the food to, again, look at all the sides and allow the enzymes to digest that food. So peristalsis, and again, this is the movement that is happening primarily in the esophagus to get you to move food from your mouth to your stomach. So it's not going to transport itself just by gravity. It needs the muscles to be acting to help this along. And so what this is it's a combination of actions between circular muscles, which are going to close areas of the esophagus to prevent backflow of the food. And so the combination of those circular muscles constricting along with longitudinal muscles, which are pushing the food down. So you can think of this as the same action that you use when you're taking chorizo out of the case. The first thing that you do is you kind of squeeze it, and then you pull it long ways to move it down. So that action in your esophagus would be called peristalsis. And so this is how food is moved from your mouth down to your stomach, at least initially, right? And so again, we have these circular muscles which are contracting. That's going to prevent the backflow of food, and the longitudinal muscles are there to help push that food forward further down the alimentary canal. So from there, after food is partially digested and it exits the stomach, it's going to go into the small intestine. And at this point, the other accessory organs that are not part of the alimentary canal become part of the digestive process. So things like the liver, which secretes bile, which is used to digest fat, or the gallbladder, which stores and regulates the release of that bile into the small intestine, become bigger players. And then also this feathery structure called the pancreas is the source of a lot of the enzymes that are going to be important in small intestine digestion. So enzymes that we're going to talk more about, like lipase, which digests lipids, amylase, which is going to digest the starches, and protease, which will digest proteins. And then finally we have the large intestine, which is where after digestion and absorption takes place in the small intestine, whatever's left over that could not be used will go into the large intestine, which absorbs that water and compacts the waste into feces so that you can get rid of the stuff that you didn't need. And so again, if we just try to quiz yourself on the different parts of the digestive system, because you will have to draw this from memory as well as know the functions. So somewhere up here in the universe, we've got the mouth. This is the esophagus, which again is moving food via that peristalsis into the stomach, which has that acidic environment, and it's killing the pathogens, and it's churning the food. And then we have things like the liver, which secretes bile. And then hiding behind this is the gallbladder, which stores and regulates that bile. So these can be taken out during surgery. You don't really need them except to digest like fats. And then sitting behind here, behind the stomach, is where we have the pancreas, which we're going to go into in excruciating detail next, which connects to the small intestines, where the majority of the digestion and absorption occurs. And then again, this is the large intestine, which is absorbing that water and compacting that waste. So now we're going to focus on what is happening in the pancreas. But before we have that conversation, let's go ahead and do a little bit of rewind on. No, I'm just kidding. Uh, so the pancreas, right? So this little guy right here, and just to reiterate where it is, it's basically hiding behind the stomach, and it's got a lot of jobs. But one of them is to produce the enzymes that are going to be used in the small intestine, specifically amylase, lipase, and your peptidases. So peptidase and protease are the same thing. They both chew protein. But before we start talking about these reactions, let's review some of what we looked at last semester about the structures of our saccharides, about the structures of our lipids, and about how we go from monomers to polymers. So just a reminder that our monosaccharides are glucose, and this is alpha glucose because this last hydroxyl group is down. We also have things like lactose, which looks a lot like glucose, except we go up at the beginning. We have our little pizza man ribose, and our dude tripping over the pizza, which is the fructose. And so these monosaccharides can be joined to form disaccharides, right? And so disaccharides means there's two monosaccharides stuck together. We had 3 of them that we learned about, including maltose, which is glucose linked to glucose. There is also sucrose, which you also know as table sugar, which is glucose linked to fructose. And then finally we had lactose, which is our only stacked disaccharide. And so this is galactose linked to glucose, and it's got this really interesting bond to it. And so in addition to the disaccharides, we had the polysaccharides. And so this were many glucose monomers linked together. And so one of them was cellulose, which causes cell wall rigidity in plants. And then we also had our starches, amylose and amylopectin. So the amylose is in the spiral form, so we have 1,4 linkages in all 3 of these. So the 1,4 linkages basically go left to right. Amylopectin is different because it's branched, and so in addition to alpha 1,4 linkages of glucose molecules, it's got alpha 1,6. And so our last polysaccharide was glycogen, which is branched of glucose molecules just like amylopectin, but glycogen is used in animals. Okay, so we're going to talk about condensation reactions and hydrolysis reactions. So this is basically the forward and reverse of I build molecules with condensation and I break them with hydrolysis. So if we go back to our monomers, right, and so say we have glucose, to join them, we do this via a condensation reaction. So I now have to find a water molecule in here, which I do by finding a hydroxyl on one side and a hydrogen on the other. And when I join them together, which a specific enzyme exists to do exactly this, they are joined via this ester link via a condensation reaction. So you can see as I join them together, the water falls off. And so this is how we go from monomers to disaccharides to polymers. And this process repeats for basically all of our biomolecules. And then just as an aside, remember that glucose is maltose together. And so again, we see these condensation reactions repeating over and over again for each of our biomolecules. So when I'm going from, say, a single amino acid to a dipeptide, right? So I'm going to start with my amino acid. Again, this is condensation because I'm joining. I find myself a water molecule. I kick it off. And then now here I've created the peptide bond for this dipeptide. And so we see this again repeatedly, including in the DNA polymerization. So I've got my phosphate, which has a hydroxyl group here. I have also my carbon number 3, which has a hydroxyl group. So again, condensation reaction, I take an OH from one side and a hydrogen from another to create a condensation reaction. And now I've got a polymer where before I only had monomers. So those are the condensation reactions where we're joining. Hydrolysis, on the other hand, is where I'm using water, so hydro, to break a bond between some larger molecule. So hydro means water, lysis means to break. So hydrolysis means I'm using water to go in there and break a bond. So this does require energy. And just like the condensation reactions, there's a specific enzyme that's going to fit with these specific substrates to allow this hydrolysis to occur. So in this case, I'm looking, I think this is, so if you look at 6-member ring, 5-member ring, that's sucrose. So if I have this linkage here, I can come in with water, and the hydrogen is going to go on the oxygen, the hydroxide will go on the other one, and then where you had a disaccharide, now you're back to a monosaccharide, right? So I go from sucrose to now glucose and fructose. Remember, this is the more complicated one. So, and we don't just see it for the carbohydrates. This is also true for protein digestion, right? So again, this is my target is the peptide bond. When I come in here with a water, the carbon is going to pick up a hydroxyl group, whereas the nitrogen will pick up a hydrogen. And so, yeah, so this is how hydrolysis works in the amino acid, and you're just going to have to take my word for it that it happens in nucleotides. Okay, so from there, we're going to focus on a specific type of hydrolysis that is conducted by the protein enzyme amylase. And so amylase is one of the enzymes that's secreted in the pancreatic cells, and amylase, as the name implies, will digest things like amylose and amylopectin. And so what we need to keep in mind here is that amylase by itself is not going to chew up an entire polysaccharide by itself, because remember, enzymes are looking for really specific shapes. So what amylase is going to do is it's going to find disaccharides and will then cut at those specific places. So the product of amylase is actually maltose. So remember, these are all glucose molecules in either glycogen or amylose or amylopectin, and amylase is going to come in and cut them off 2 at a time to create maltose. So this maltose cannot be directly absorbed by our cells, so it needs to be further degraded. And so maltose, as you can guess, is further broken down by an enzyme called maltase. Again, because it's looking for those disaccharides. So maltase is going to grab hold of those maltoses and then break them down into 2 glucose molecules, which can then be absorbed. So that's how starch digestion and absorption is going to start. I want to also remind you about triglycerides, right? So triglycerides are not 3 glycerols. Triglycerides is 1 glycerol molecule connected to 3 different fatty acids. So this is what we colloquially refer to as fat. This is what is stored in our midsection. And then just a reminder that a fat molecule can store about 460 times more energy than sugar. So gram for gram, this is a more efficient way for animals to store energy. And again, it is the condensation reaction between a glycerol right here, so we've got my hydroxides, and 3 different fatty acids that are coming together via those condensation reactions to get you a triglyceride. And so just like for our other reactions, right, we build it through condensation and then we break it through hydrolysis. So I have these same molecules, I come in here and I basically reattach the hydroxyls on one side and the hydrogens on the other to go back, right? And so the enzyme that does this is lipase. And again, that lipase is coming from the pancreas. So I also have peptidases or exopeptidases or proteases, whatever you want to call them. So these are the ones that are attacking that peptide bond. And again, those enzymes are made by the pancreas. So once they're created in the pancreas, all of these enzymes are secreted into the small intestine. And so in addition to the lipase, which is digesting those triglycerides into fatty acids, and the amylase, which is breaking down those starches, you have these proteases. You also have nucleases, right? Because stuff that you're eating probably has nucleotides coming in, and there's specific enzymes to break that down as well. And so if we take a step back, right, and we look at animal cell structure, we know that all cells have the plasma membrane. We all have cytosol. Um, the easy organelle to point out is always the nucleus. And then we've got mitochondria, which do respiration and are responsible for energy generation. You also have things like the ribosomes, right? And in the case of a eukaryotic cell, it's the ADS ribosomes, and they come in two flavors, the ones that are free versus the ones that are bound to the rough endoplasmic reticulum, which will then pass proteins to the Golgi. And and then the Golgi will package those proteins into large vesicles. And so what I want to focus on is how we get proteins out of the cell, right? Because I've got my pancreas who are creating loads of enzymes for me, which are just a kind of protein, and secreting them into the small intestine. And so if we focus for a minute on where exactly protein is coming from, we've got two sources. One of them are the free ADS ribosomes, and the other, or the bound rough endoplasmic reticulum ADS ribosomes. And these are destined for two different places, right? The ones that are free in the cytosol are synthesizing proteins that are going to stay inside the cell, versus the ones that are attached to the rough ER and are then going to be transported to the Golgi and vesicles are proteins that are going to be secreted. They're going to go outside. And so just a reminder of what these look like, right? So we've got our nucleus here, and the rough ER is always like directly attached to that nucleus, and then we've got vesicles kind of going out. So these vesicles are full of proteins that are being secreted to the outside, outside that cell. And so we had a special name for this kind of transport for where proteins are exiting, and we call that exocytosis, right? So this is where proteins are exiting. So they're being created on the rough ER, being passed to the Golgi, where they're getting modified with sugars and then repackaged into vesicles, which then go outside the cell. So this was a type of active transport that did require ATP energy, right? So again, we start with our ADS ribosomes. They're creating those enzymes or proteins in the rough ER. They package them into vesicles first, then send them over to the Golgi apparatus, which then modifies them, usually by adding sugars. And once it's done with that, it repackages them and then sends them to the membrane, where those vesicles fuse, and the proteins that it made are free to move about kind of like the country after that point, right? Okay, so I told you that story so that we could come back and tell you this one. So we're back to the pancreas, right? This little feathery organism. And it is directly attached to the small intestine, which makes sense because it's making the enzymes that are going to go in there. But it's also got other regions for other important tasks. So if I take a closer look at the structure of the pancreas, we actually have two kind of gland centers in here, an exocrine gland and an endocrine gland. So the exocrine gland is what is going to be secreting those enzymes that it's making into the small intestine. So that's one thing. So our enzymes like lipase, amylase, and protease. But then it also has endocrine gland. And remember, endocrine is all about hormones. And so the most important hormone that the pancreas is creating is actually insulin, which is produced in these beta isolate cells in the islets of Langerhans here. And that insulin will go all over the body to say, hey, you've got glucose in your blood, go ahead and take up that sugar. And so where these are located really matters, right? Because the exocrine, again, I'm making enzymes to go to the small intestine, so I'm connected to a duct that's going to secrete to the small intestine. Whereas in the endocrine glands, I'm connected to capillaries because that insulin needs to go all over the body, right? So if we take a closer look, right, so we're here now in the exocrine gland. These cells are very specialized. So I'm secreting enzymes, which are proteins, that are going to go to the small intestine. So thing one is I'm attached to a duct, which is going to dump straight into the small intestine. And I have these very specialized secretory cells. And what is special about them is if my job is to secrete and to make enzymes, then it would make sense that my structure, I'm going to be very heavy on a rough ER. I'll have a lot of that around, and I also should have a lot of vesicles, right? Because my whole job is to make proteins, these enzymes that are going out. And that is definitely what we see, right? When we look at these cells under micrographs, is that you have these huge areas of rough ER and really large vesicles, because that's their whole job, is to create these vesicles to then secrete into that duct to go to the small intestines. That's their whole job. So that's how the exocrine gland works. If we look at endocrine, again, we're switching here. So endocrine is about hormones, and the endocrine cells are in the beta islet cells, and these are snuggled up against capillaries, right, against blood vessels. And so they're secreting that hormone, and then instead of going to the small intestines, now they're secreting that hormone to go all over the body. And so once these endocrine cells make that insulin, it is transported to the blood vessels, and then it can go all over the body. And once insulin is traveling all over the body, it can come out, activate insulin receptors, which tell the cell, hey, open your glucose channels. There's sugar out here for you to get. So that's the long story of pancreas and how they work and their role in nutrition and digestion. And so let's look at another question, right? So why can't we eat grass? Like, why is it that bunny rabbits think that grass is the most delicious thing ever, and so do cows? And that technically speaking, right, cellulose is just a bunch of glucose molecules. So why are we not able to like enjoy grass? Like you eat it and you think it's the most disgusting thing ever. And the reason is that humans cannot digest grass, which contains a lot of cellulose, because we do not have access to the enzyme which degrades it, which is cellulase. So the cows and the bunny rabbits do have bacteria in their gut that makes that enzyme, which helps them break down that grass, right? So if we don't have it, we can't eat grass because we can't digest that grass, right? So that is why humans, we can't be like little goats and go like, cut down our own yard. Okay. So we focused a lot on the pancreas, which again, if I'm, it's right here and it's usually hiding behind the stomach. Let's look at some structures that we're going to see in the intestine itself. So if I look at the intestine, right, I have a lot of layers here because of course nothing is ever just simple. And if you've ever eaten like tripe, right, you know that when you cut open those intestines, that there's all these little like finger-like projections on the inside. And so these are actually something called villi. And so we have a lot of structures in the intestine itself. We've got muscle layers, which again, we need to help move the food along. On the inside, we've got the villi, and then this whole structure is called the mucosa. And then underneath that we have the submucosa. So this is like the longitudinal section here. And if I look at like a small, like a up-close version of this, I can see these villi that are here. The entire thing is called the mucosa, and it is sitting on top of the submucosa, and located in here are the muscles, right? Those circular and longitudinal muscles that are helping to move that food along. So let's focus a little bit more on the structure and function of these villi here. So again, we're just looking at a larger section of the small intestine, and now we're going to kind of zoom in and look at the structure of the villi. So these guys here. So there are basically three main parts, right? So we'll start with like the largest one. So the one in the center is called the lacteal. And so this is going to connect to lymphatic vessels, and it's going to matter, who absorbs what. And so for this, the lacteal is what absorbs the lipids, your fatty acids, whereas the blood capillaries are where the absorption of glucose and the amino acids is occurring. And we'll talk in excruciating detail later about how these are actually moved across the membranes. And then this villi is also covered by a thin epithelium at the top, of which are microvilli on top. And so those are there to, again, increase areas for surface absorption. So just like, let's take another look that we have this villi. Again, the important parts are your epithelium, which is on the outside here, which has microvilli on top of it. You have your lacteal running down the center, which is where we have things like fatty acids and the glycerol. being absorbed, and then the capillaries is where glucose and your amino acids that were digested either by amylase or the proteases are going to come in. So that's what we're going to focus on for right now, those structures, because I'm sure you're tired of listening about digestive system. All right, so we'll see you guys next time.